The next speaker is Clément Cochain, who is from the Comprehensive Heart Failure Center Würzburg, and he will give a talk on time series single-cell analysis of myeloid cells after acute myocardial infarction in mice.
Thank you very much. I'm really happy to participate to this very nice meeting, and I'm going to present you some very recent data that we obtained about single-cell analysis of cardiac myeloid cells in the context of acute myocardial infarction. So this work is very much a cooperative effort between my team, where we are not at all computational biologists. And for all these single-cell stories, we work closely with the lab of Emmanuel Saliba in the Helmholtz Institute in Würzburg, and in particular with Ezan, that is here today and performed a lot of the analysis that I'm going to talk about. So I'm sure that most of you have a pretty good notion of what a myocardial infarction is. So the heart is basically a muscle that contracts and pumps blood throughout your body. And this organ itself is very much dependent on the blood supply that it gets from a network of coronary arteries. When one of these arteries gets occluded, most of the time due to atherosclerosis, then the downstream tissue that normally gets blood from this artery will find itself in a state of ischemia, so does not receive any more oxygen or nutrients. And the cardiac muscle cells, so the cardiomyocytes, will die. And unfortunately, these cells have almost no capacity to self-renew, so what will happen is that there will be no regeneration of this tissue, but a process of cardiac repair that leads to the formation of a fibrous scar. So here, for example, in adult mice or in a human heart. So this poses a number of problems. So this scar cannot contract, so the function of the organ itself is affected. In the long term, there will be a process of remodeling of the heart, which leads to heart failure, and also this fibrosis favors the appearance of arrhythmias, such as ventricular fibrillation and sudden cardiac death. So what happens in the heart is basically a wound healing process, and this wound healing process is very much dependent on the inflammatory response. So immediately after infarction, there will be a massive recruitment of various immune cell types, so mostly cells of the innate immune system, such as monocyte and neutrophil. And here, this is a very simplified picture, but these cells are on one end necessary for this tissue repair process, but on the other end can also promote tissue damage. So on top of this functional heterogeneity of these cells, this infiltration process of this different cell type is very dynamic after infarction. So this is what we know from a mouse model, that in the steady state, the heart already contains different types of cardiac-resistant macrophages. And immediately after infarction, this cell will mostly vanish, or at least be overwhelmed by recruited cells, so neutrophils. And these classical monocytes, so the so-called Ly6Ci or plus monocyte, that are called this way because they express this Ly6C marker. We'll talk a lot about these cells during the presentation. If you move it further, like day 3 after infarction in mice, these classical monocytes will become prevalent, and then at later time points, they will differentiate into macrophages. If you move even later in time, these monocyte-derived macrophages can persist, and these cardiac-resistant macrophages can reappear via self-renewal. And all these different phases of cell infiltration more or less correspond to overlapping phases of the repair process in the tissue, with first like removal of all the dead cells, so cleaning up of the tissue, and then neovascularization and formation of a mature scar. So as you can see from this small introduction, single-cell methods are super attractive to us to kind of capture the whole complexity, the heterogeneous function of this cell, and also this dynamic of cell infiltration. So to some extent, some papers have already used single-cell methods to address this question. So these are papers from the last few years that are very interesting, but one major limit, at least from my point of view, is that all these papers looked at a single time point of the disease. So 2 papers looking at 4 days after infarction, 1 paper looking at 11 days. And what we tried to do in this recent project is to focus more on the dynamic of this innate immune response in the acute phase after infarction, with some specific questions, such as is there some heterogeneity in these classical monocytes or in neutrophils? We also wanted to focus on the monocyte to macrophage transition, and also a bit on what could be the functional impact of the differentiated macrophage heterogeneity in the heart. So to this end, what we did is pretty simple. We used the model of myocardial infarction in mice and collected hearts before infarction or at various time points after induction of infarction, so 1, 3, 5, and 7 days. And from this heart, we sorted total viable cardiac leukocytes and processed them for single-cell RNA-seq using the 10x pipeline. And we'll focus here only on myeloid cell data. So we kind of put all the data together and could trace back the time point of origin of the cells. So with this first data set, what we could recapitulate is obviously the expected kinetics of inflammatory cell infiltration within the heart. And what we saw in this, this first experiment was time-dependent heterogeneity of classical monocytes that are around here on this map, and here the time points corresponding. And yeah, these very early time points, we could see kind of heterogeneity of these monocytes and also of neutrophils. So some of these data are a bit surprising to us. So we wondered whether this heterogeneity could also be due to technical variability between samples. So we did very recently a kind of validation experiment where here we try to get rid of most of the bias that can be a problem in these models. So what we did is from 3 early time points of the DC, so 1, 3, and 5 days, we obtained all the cells, all the myeloid cells in a single sort, and then labeled them with cell hashing antibodies and also a few site-sec antibodies, and then pull the cells again for a multiplex run, once again using the 10x pipeline. So I won't talk about neutrophils anymore, but we could kind of confirm our first data and time-dependent heterogeneity of these cells, and now focus more on what happens to monocytes and macrophages. So here are some of the data that we got. So with the hashtag signal, we can pretty nicely trace back the time point of origin of the cell. So the ones coming from 1 day after infarction, 3 days, and 5 days. And looking at monocyte and macrophage marker genes, we can also capture kind of this transition from monocyte in the first time points towards macrophages at 5 days after infarction. And what we could already see is that in these various clusters that correspond to monocyte according to their transcriptional profile, we can see again kind of time-dependent heterogeneity. So we went a bit further into that, and specifically into the heterogeneity of these classical Ly6C plus monocytes. So according to our site-sec data, at least 3 of these populations of these clusters that we found in this experiment correspond to Ly6C plus monocytes. And we can see the one that we find from the day 1 time point, from the earliest time point, are pretty much different from the others. So here they will express more chemokines, also VEGF, which is a pro-angiogenic factor that promotes neovascularization. And we move a bit later in time, so 3 and 5 days after infarction, we find what I would call standard Ly6C plus monocyte, classical monocytes, which are more or less the same that you can find in the steady state in the blood, bone marrow, or spleen. And we also find type of classical monocytes with a strong signature of type 1 interferon response. And what was also important is that this data that we saw here, if we go back to the first data set we generated, we see more or less the same gene expression patterns and the same time-dependent heterogeneity in these cells. So here with this experiment, we kind of refined the picture that we had before, and can see that actually these cells that will all be classified as classical monocytes are quite transcriptionally heterogeneous at these different time points. And one of question that we had is that could these cells then differentiate into different kind of macrophages? So this is very much a work in progress, and we are still trying to figure out what is the best way to address this question and reconstruct cell trajectories. One thing that we tried recently, on our first data set, we tried to apply the RNA velocity analysis on this data set. So it's a bit difficult to interpret, but yeah, altogether it seems to make kind of sense time-wise and with the differentiation pathway of monocyte to macrophages. But as I said, this is clearly very much a work in progress. What happens at least is that we can see monocyte turning into macrophages, so different type of macrophages with quite specific enriched genes in this different subset. And using a data set that were already published, we can confirm that some of these cells we find even at later time points. So our data set is from 1 to 7 days. And lots of these cells we can find also at a later time points after infarction. And I will focus now a little bit on one specific type of macrophage that we found in this data set that I name here after 2 of the most highly differentially expressed genes in this cluster. And so this macrophage seem to have, at least if we just look at transcriptional data, this will need to be confirmed with functional studies, seem to have a pro-healing phenotype. So they express a lot of genes that have been previously associated with tissue repair in models of myocardial infarction, such as this GF15, IGF1, or TIM2. And what was also a bit interesting for us when looking at gene expression patterns in these cells is that this specific macrophage subset had a lot of similarities with a macrophage subset that we observed previously, but in a completely different context. So in the context of atherosclerosis. So this is a pretty rough comparison, and we clearly check that a lot of enriched genes in this cell, so either in the heart, in our data set, in another data set, and in data from atherosclerotic aorta in mice, they have a lot of similarities in their gene expression profile. And we'll try to go a step further and check whether these cells could have upstream regulators that will be behind this gene expression pattern. And I'm not going to show everything, but we apply this CYNIC program to identify activated gene regulatory network in these data sets. And one interesting hit that we had is this regulon, this NR1H3 regulon, which encodes the liver X receptor alpha. And so going a bit beyond this cardiovascular disease context, we also used a recent data set, so that was published a few weeks ago in a model of skin wound healing. And when reanalyzing the immune cell data subset in this model, we also found cells with a lot of similarities in there. And if you go even a bit further, even though it seems a bit far-fetched, if you look at microglia in the brain in neurodegeneration model, there seem to be a lot of similarities, at least on the transcriptional level. And so, yeah, this is, of course, a very rough comparison, and so it will be interesting for us to have tools to really integrate these data sets together and get a bit more precise information. So, yeah, so in conclusions, this work is very much the beginning of, yeah, there remain a lot of things to do for us. And the first thing that we would like to do is move upstream in time within the tissue, and also upstream in the organs where the myeloid cells are produced, to check what happens in this myocardial infarction situation, for example, in the bone marrow or in the spleen, where these monocytes and neutrophils can be produced. So, of course, for us, this kind of data is very nice, but it's very descriptive, and what interests us in the end is to have a notion of what would be the functional impact of all what we observe, and that's also what we plan to do in the near future. Spatial localization method would, of course, be of great interest to us. And one last thing, it's always necessary to remember that all we do here is in one strain of laboratory mice. So clearly, we would like to move in the near future towards more human studies, but as was said in the previous talk, this is easier said than done. But at least we have access to a lot of samples in the Heart Failure Biobank in Würzburg, and we will have to check what can be usable in this biobank. So thank you for your attention. I will be happy to take your questions and feedback.
That was a very nice presentation. So now I was wondering, when the monocytes differentiate into the macrophages via the different stages and then secrete paracrine factors like GDF15, IGF-1, did you actually look also at neighboring cells, what that brings with it? So is it really a regenerative signal? So is the inflammation in the beginning good, and then if you have it chronic, is it bad? So can you measure the effects on cardiomyocytes or cell type next door?
This is, of course, a very interesting question, and but that's quite difficult to address. But of course, the basic notion that we have is that we cannot categorize inflammation as good or bad. It's not such a clear dichotomy. But that's why we also do this kind of studies, to try and find out the way to target this specific type of macrophage or this specific type of macrophage. And then we have, that's what we do basically in my lab. We have functional models to see how the heart recovers after infarction. But yeah, that's clearly what we are going towards in the future. Yeah.
Thank you, very nice talk. I just was wondering about the macrophage activation from skin to heart versus microglia in the brain. Have you tried to compare them? What are the tissue-specific signatures?
No, not yet, but of course that will need to be done, and that's why this is a more or less a random observation of just putting one data set next to the other, but that's why we need to integrate data sets together and see what are the specifics of macrophage in each tissue, clearly.
Maybe I have a more technical question. So you had this initial data set where you said that clusters are more driven by maybe batch, and then you have this new data set where you did the hashing, and then you see less of this technical variation. Did you ever compare and find out what are actually the genes that drive the different clusters? Like, what were your technical biased genes in the first data set?
So first of all, the nice thing is that when we take the second data set and go back to the first one, whether for the monocyte macrophages or for the neutrophils, we kind of see the same populations. But what we have in the first data set, and that kind of drives more heterogeneity, it's just an hypothesis so far, but it has to do with the way the samples were prepared and different ambient RNA contamination in the samples from the first data set that we don't have in the second one, actually.
Okay. Any more questions? Yes.
Maybe a short note on the time series. So I think it's really cool that you get those out, but we just had discussion about, let's say, differential expression analysis, but in particular those velocities. I'm not sure if the model is to be used for the sort of integrated data across the time series.
Yeah, I'm not sure either, actually. That's why we're still trying to figure out how to best analyze the data we have now. And yeah.
So I guess I would do it just per time point, essentially.
Sorry?
What I'm saying is I would just do it per time point.
Yeah, each time point separately.
Yeah,
yeah, that's also something that Ezan thought about, but that we did not do so far. But yeah,
and that's how it has been proposed, right? So I think you actually need to extend the model.
Yeah, thanks.